genetic engineering allows inserting specific genes, of one species into another, creating transgenetic animals, but this only adds isolated traits, but not a full species change, but inside cloning. Via somatic cell nuclear transfer replicates exact genetic copy within the same type of specimen bias, that is only one species of organism, meaning to say that we call it cloning, but it remains unaltered within the whole species. In general, transgenetic transformation is illegal according to FDA, but cross-species genetic engineering inside animal found to be ethical, but is against big novelty towards animals. Understanding this genetic process begins with specific terminology that describes the units of heredity and their expressions. Genes are basic expressions of this heredity and carrying the whole instructions of the organism's traits. Different versions of genes are called alleles. For example, color of a flower might contain a gene of flower as allele for purple and different allele for. These alleles can be both dominant and recessive one. Dominant one expresses its trait only if one copy present, and recessive one only expresses its traits for more than 2 copies. A specific combination of alleles an organism possesses for a particular trait of the organism is its genotype. An observable physical traits that results from this genotype is called its phenotype, whereas an organism with 2 common alleles for a single trait are called homozygous. About genetic crosses is wholly different from cross species genetics of full species change or cloning. On the other hand, ERVs, endogenous retroviruses sequences, are nothing but molecular fossils that reveals ancient species evolutionary dynamics integrated into specifically animal genomes. Their presence across species highlights past retroviral infections that presents in the current situation as a fossil, but that jump into the hosts of these genomes, specifically animal genomes. That's why specifically whenever any animal species found to be in the danger of vanishing, their ERVs are considered as one of the medium of influencing planetary genetic evolutions across whole species present in the present environment. These ERVs normally generated from germline cells infected through infected organisms, allowing their DNA to persist inside vertebrates as only remnants, unlike active virus. For instance, inside bats and cats, similar ERVs shows different fates, like retranspositions in cats versus reinfections in bats. Hence, these ERVs are nothing but raw genetic material as envelope proteins for new functions inside big laboratories.